Welcome to the deep dive. Today we're diving straight into something fundamental,
Yeah, really fundamental. How our brain cells actually talk to each other.
using these tiny chemical messengers, neurotransmitters.
Exactly. And we've got some great sources looking into these, well, microscopic communicators. We're focusing today on how they really shape your memory, your cognitive abilities,
It's kind of mind-boggling, isn't it? Billions of neurons, trillions of
all
connections.
happening constantly. It's this incredibly intricate chemical dance.
Right. So our mission here is to give you a clear picture of neurotransmission, why it matters so much, but, without making it feel like a textbook lecture.
Let's do it.
Okay, let's unpack the basics then. How does this message sending actually work?
So it kicks off with an electrical signal. That's the action potential. It travels down the neuron's long arm, the axon.
Okay.
And when that pulse hits the very end, these structures called terminal buttons,
Uh-huh.
it triggers the release of neurotransmitters. These chemicals then spill out into this incredibly tiny gap.
The synapse.
The synapse, exactly. We're talking about 1/1,000,000th of a centimeter wide. It's
Oh.
minuscule.
Wow. So these neurotransmitters are like tiny notes passed between cells.
That's a really good way to put it. They're the body's own chemical messengers. They float across that synapse,
Right.
and they dock onto specific receptor sites on the next neuron, the postsynaptic membrane.
And it's specific, right? Like
It-
a key.
Precisely, often called a key in a lock mechanism.
Yep.
Each neurotransmitter key fits only certain receptor locks. And some keys might fit better or turn the lock more effectively than others.
Okay, so the message gets delivered. What then? Do these chemicals just hang out in the gap?
No, the brain's very tidy, actually. Once the signal's sent, the neurotransmitters are cleared out pretty quickly.
Ow.
Two main ways. Either enzymes in the synapse break them down, just neutralize them,
Oh.
or they get sucked back up into the neuron that sent them. It's called reuptake.
Recycling them.
Yeah,
basically.
exactly.
They-
It cleans the slate for the next message. Super efficient.
And these tiny, tiny events, they control so much.
Absolutely everything. I mean, neurotransmission underlies your mood, your sleep, learning, memory, pleasure, focus. Even things like mental illness are often linked to imbalances here. It's the bedrock of thought, feeling, action.
The sources you shared highlighted a few key players. Let's maybe zoom in on some specific ones. Acetylcholine came up a lot.
Yes, acetylcholine is a big one. It's really well known for its role in memory, especially memory consolidation, making memories stick.
Where does that happen mainly?
A key area is the hippocampus. There's a classic study, Rogers and Kesner, showing how crucial acetylcholine is for getting new information into long-term storage there.
Okay, and dopamine. That's the pleasure one.
Often called that, yeah. Dopamine drives the brain's reward and pleasure centers. Super important for motivation. And interestingly, disruptions in dopamine are linked to addiction, but also things like Parkinson's disease.
Then there was norepinephrine and serotonin. What about those?
Norepinephrine is mainly about arousal, alertness, kind of gets you ready for action. Serotonin is more involved in regulating sleep, general arousal levels, and importantly, emotion and mood.
So norepinephrine is like the go signal, and serotonin helps manage mood and rest.
That's a decent way to think about it, yeah.
And you mentioned GABA earlier, saying it was different.
Yes, GABA. Gamma-aminobutyric acid. It's the main inhibitory neurotransmitter. And this difference between excitatory and inhibitory is really fundamental.
Okay, explain that. Excitatory versus inhibitory.
Sure. Excitatory neurotransmitters, like acetylcholine we mentioned, make the receiving neuron more likely to fire its own electrical signal. They sort of nudge it towards action. It's called depolarization.
Makes it more positive inside.
Exactly. While inhibitory ones, like GABA, do the opposite. They make the neuron less likely to fire. They hypopolarize it, make it more negative inside.
So they put the brakes on.
Precisely. It's this constant push and pull, this balance between go signals and stop signals that allows the brain to process information so effectively.
It's like a complex traffic light system. Okay, let's go back to acetylcholine and memory. You mentioned that Rogers and Kesner study.
Right, from 2003. It's a really neat study using rats to look at acetylcholine's role in memory.
What do they actually do?
They had two groups of rats. One group got scopolamine, that's a drug that blocks acetylcholine receptors, basically stops acetylcholine from working properly.
An antagonist.
Exactly, an antagonist.
Right.
The other group got a placebo, just a saline injection, so no active drug. This is the control group.
Important control.
Crucial. Then both groups were put in a maze they'd already learned, where they knew food was located. The researchers watched how long it took them to find the food and how many wrong turns they made.
And the results?
The scopolamine rats, the ones with blocked acetylcholine, were much slower and made way more errors.
Ah,
The control rats, with normal acetylcholine function, remembered the route much better, found the food faster, fewer mistakes.
so blocking acetylcholine messed up their memory retrieval.
Strongly suggested that, yes. It pointed clearly to acetylcholine being important for recalling that learned information.
The source mentions strengths and weaknesses, though.
Always important to consider. A strength was the clear design, pretty easy to replicate, and the control group isolating the drug's effect.
Right.
The main limitation, of course, is generalizability. It's rats. Can we be sure it works the same in humans?
A classic issue.
But, and this is where it gets compelling, later research in humans backed it up.
Okay. Tell me about that.
Well, there's a study by Antonova and colleagues from 2011. They used fMRI.
Brain scanning.
Exactly. Watching the brain in action. They got healthy young men, gave some scopolamine, some placebo, double-blind. So the participants or researchers knew who got what initially.
Good setup.
Then, while in the scanner, the guys played this virtual reality game, the Arena Task, tested their spatial memory learning where things are.
What do they have to do?
They learned where a pole was in this virtual arena. Then the screen went blank. They had to mentally rehearse the route. Then the arena reappeared, but they started somewhere new and had to find the pole again.
Tricky. And the fMRI showed.
When the guys were on scopolamine, their hippocampus activity was significantly lower during the learning part of the task.
The hippocampus again. Same area as in the rats.
Exactly. And that lower activity directly linked to them doing worse on the memory task. So, pretty strong human evidence that acetylcholine is key for encoding spatial memories.
That really does tie it together nicely. Okay, let's switch gears to GABA, the main inhibitory one. How does it work?
Right. GABA's job is basically to calm things down, to inhibit neural firing. It does this across the brain, including the hippocampus and the frontal lobe, which is crucial for, planning, decision-making, higher thought.
And this, in addition, this calming down actually helps thinking. Seems a bit backward.
It does seem counterintuitive at first, but think about it. Our brains are constantly buzzing with activity. GABA helps filter out the noise. By dampening down irrelevant signals, it helps you focus, concentrate on what matters. It essentially increases your cognitive capacity, your working memory, by reducing interference, like noise cancellation for the brain.
That makes sense. Reducing the static. You mentioned a study on GABA in older adults.
Yeah.
Porges it all.
Yes, 2017. They looked at GABA levels in the frontal lobes of older adults who didn't have dementia. They measured the GABA concentration and also gave them a standard cognitive test, the Montreal Cognitive Assessment.
And what was the link?
A pretty clear positive correlation. Older adults with higher GABA levels in that frontal area performed better on the cognitive tests.
Interesting. So more GABA, better thinking in older age.
But the findings suggest that yes, and it's significant because GABA levels do tend to drop as we age naturally. This might be one reason for some age-related cognitive decline.
So maybe boosting GABA could be helpful.
That's definitely an avenue researchers are exploring. Potential treatments for age-related issues, maybe even dementia prevention. Some newer studies like Prevost et al. In 2019 are looking right into that.
This brings us back to those terms, agonists and antagonists. Can you just recap those in this context?
Sure. So agonists are chemicals that bind to a receptor and activate it. They mimic or boost the natural neurotransmitters effect.
Like acetylcholine itself.
Exactly. Acetylcholine is an endogenous agonist made inside the body. It binds, activates the receptor, message sent.
Got it. And antagonists.
Antagonists also bind to the receptor, but they block it. They sit in the lock, but don't turn the key, and they prevent the real key, the neurotransmitter, from getting in.
Like scopolamine blocking acetylcholine.
Precisely. Scopolamine is an acetylcholine antagonist. It reduces acetylcholine's effect, hence the memory issues we saw in the studies.
And you mentioned drugs can be agonists, too, from outside the body.
Right, exogenous agonists. Nicotine is a classic example. It acts as an agonist at certain acetylcholine receptors.
Is that why people feel more focused sometimes when they smoke?
That's part of the mechanism, yes. Short-term boost in alertness or focus for some, but, and this is crucial, the long-term negatives, including on memory and health, are really significant. Caffeine is another common one, acting on adenosine receptors.
So that Rogers and Kesner study really shows both sides, doesn't it? The normal agonist effect of acetylcholine and the blocking antagonist effect of scopolamine.
It's a perfect illustration, yeah. You see the natural agonist working in the control group, and you see what happens when you introduce an antagonist that disrupts it. Understanding agonists and antagonists is key to understanding how so many drugs, medicinal or otherwise, affect our brains.
This has been incredibly clear, thank you. But the source material also had a really important warning, didn't it? About being too simplistic.
Absolutely. A caution against reductionism,
Yeah.
saying behavior is just neurotransmitters.
Right.
It raises big questions. Can something as complex, as profound as falling in love really be reduced to a love cocktail of chemicals like dopamine?
Or is feeling down during winter just about serotonin levels, it feels like? More complicated than that.
And that's really the mainstream view in the field now. Neurotransmitters are undeniably crucial, foundational, but they don't tell the whole story.
There are other factors.
So many other factors, our genetics, our life experiences, our environment, learning, social interactions. It's this incredibly complex interplay. Neurotransmitters are vital cogs in the machine, but they aren't the whole machine.
So, wrapping up this deep dive, we've covered a lot. The basics of how neurons communicate, this amazing chemical messaging system.
Yeah, focusing particularly on acetylcholine's role in memory, backed up by both animal and human studies.
And GABA's role in cognitive function, especially its inhibitory action, helping us manage thought processes, potentially linking to aging.
And we clarified those key concepts. Agonists mimicking neurotransmitters, antagonists blocking them.
But always with that final important reminder, behavior is complex. Neurotransmitters are part of a much bigger picture.
Definitely. And maybe a final thought for you, the listener, to chew on. Considering this delicate chemical balance we've discussed, what does that suggest about why we see such huge individual differences in how easily people learn, in their moods, their vulnerability to certain conditions?
It really highlights how intricate we are.
It truly does. It's a fascinating area, and we're learning more all the time.